Hello, everybody. We are in week 5. You probably already know you have an exam to take. There is an announcement that goes through how to study for that exam and what to expect from the exam. For those of you in the online lab, you also have an online lab practical, and in the announcement, it talks about how to get ready for that as well. So let's dive into week 5, chapter 5, viruses and prions. Now, again, I kind of want to talk about why is this one of my favorite things? Simply because we want to think about adaptation and natural selection. How did these things get to be the way they are? Well, the answer is simple. They had those traits that allowed them to survive and reproduce. Okay, so let's get on with the topic. All right, discovery of viruses. I don't go into a lot of history in this class, but essentially one way that scientists had originally studied bacteria was by filtering out the bacteria when it was in infectious fluids. So the researchers here took, essentially you see the leaves in the lower left-hand side of the slide, and these light yellow areas or pale green areas are actually areas where there is a virus called the tobacco mosaic virus. When I was in graduate school, I did not smoke, but people who smoked were always asked to wash their hands thoroughly before they handled any plants, so they weren't potentially infecting those plants. Okay, so viruses, when these researchers attempted to essentially take that host tissue and separate it through porcelain filters that normally would trap the bacteria, they found the infectious material still remained after being filtered. Okay, so what they did is they took that filtrate and then applied it back to plant material and saw that it continued to cause or caused infection in healthy plants. Okay, so interesting. So like if you decide that you really are keen about science and research, this is the kind of stuff that you look to solve in problems, solve in labs, excuse me. On the right-hand side, we see an electron micrograph of an actual tobacco mosaic virus, and you see that they look like long, stiff rods. So we're going to talk a little bit about this as we get into the structure of viruses. What's super important to realize is that when we're talking about viruses, and then also prions, which we're going to get into, we're talking about teeny, 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 tiny things, okay? So if you take a look at this diagram from your textbook, they show us that viruses range from 20 nanometers to 1,000 nanometers in diameter. So you're really talking about the small end of the spectrum of size, okay? And in this figure, we see a yeast cell, likely budding, and we can see how large it is. This is a eukaryotic cell. And then, of course, in the upper right-hand corner, we see E. coli, which is a rather large bacterium in the big scheme of things. For those of you in the on-campus lab, you probably didn't think they were very big when you were looking at them, even magnified 1,000 times, but indeed, they're about 2 microns long. So you get an idea here of scale. All right, so we use light microscopes, these are the traditional microscopes you would have used in a high school setting or at the college setting. And if you decide to go into virology, or you're going to do cell ultrastructure research in graduate school, then you would use an electron microscope. All right, so viruses, let's just put this out there. The properties of viruses do not include the properties that would make them alive. So it's kind of an odd thing. That's the first thing that we say. Viruses are not living. So in a minute, I will talk about what does it mean to be alive. But essentially, if we go through this list, the very first thing, they are obligate intracellular parasites of nearly everything else. Bacteria, protozoa, fungi, algae, plants, and animals. Okay, so they have to have a host. If they don't have the host, they can't survive. So that right there, can't be alive if you can't live on your own at all, ever, and you can't reproduce yourself on your own. It's estimated that there are 10 to the 31st power virus particles on Earth, approximately 10 times more than the number of bacteria and archaea combined. So they are ubiquitous. There are so many of them. They're in all major environments on the Earth, and they've had a major impact on life. Okay, we already talked about them being ultramicroscopic, so teeny, teeny, tiny. Their structure is compact and economical, but it is not considered a cell. So we do need to talk about the structures that make a virus a virus. The next one we already talked about. They do not independently fulfill the properties of life. So they're not considered to be alive, but they affect life. In fact, they are, again, obligate parasites of living things so that they can do all the things that they need to do to propagate or replicate themselves. They have a basic structure, consists of a protein shell, we call it capsid, around a nucleic acid core. That nucleic acid core is DNA or RNA, never both. Okay, so they do have some rudimentary genetic material that codes for what they need the host cell to do. You can see the nucleic acid can be double-stranded or single-stranded, DNA and RNA. That's kind of outside the scope of what we need to talk about. Next one, molecules on the virus surface give them high specificity for attachment to the host cell. Viruses are very, very specific. So a rhinovirus, a virus in your nose, is not going to attack your gastrointestinal tract, right? Like norovirus. So they're very, very specific to the host cell that they need to invade, which also means that their host organism is also very specific. Getting back to this idea of not being alive, viruses cannot replicate themselves on their own. They do that by taking charge of and control of the host cell's genetic machinery, and then they guide their own, what we call synthesis and assembly of themselves to make more. Last two things, they lack most enzymes that you need for metabolic processes. So when we think about metabolism, we're thinking about all these chemical interactions that are keeping us alive, and they can't do that on their own, all right? And lastly, they most of them are lacking machinery for synthesizing proteins, so that's why they need a host cell. All right, I do believe this is just a repeat. So if you like this a little clearer, here it is. One thing on this slide, they have relatively few genes, but what they do have, it is enough to guide what they need to happen. Okay. All right, so this is a figure not from your textbook, but it shows us those properties that do define living things. And taken in totality, this defines living things. So we have growth and development. We can reproduce on our own, meaning we don't need a host cell. Yes, we know there's asexual reproduction and sexual reproduction, and in bacteria we know there's conjugation. That's still what we would call reproduction. Heredity of traits, so we pass down our traits, and we don't need other machinery outside of ourselves to do that process. Homeostasis, maintaining the stable internal environment, that's super important for living things, not for viruses. Metabolism, we know that viruses will be hijacking their host and having the host essentially help with their metabolism. They are acellular in nature. They are not cells. They have a completely different structure. And we know that living things respond to the environment. So this kind of goes hand in hand with homeostasis. You have to be able to respond to make sure that you're staying in that, happy medium we call homeostasis. All right. So a quick Venn diagram to show you how bacteria and viruses are different and then how they are similar. So bacteria and viruses both can cause disease, right? They are both spread by bodily fluids in close contact with infected people, animals, so on. We know that neither one of them are going to have a nucleus, but we know that the bacteria is going to have that plasmid and that nucleoid. And viruses are going to have DNA or RNA, just enough to hijack the host cell and then replicate itself. Bacteria respond to antibiotics. So essentially, those are medications that target that bacterial cell. And viruses respond to antivirals. There's not a lot of antivirals out there, but certainly there are a handful. All right, so you can stop on this if you want to and read a little bit more. So I'm going to move on. All right, so it's really always awesome to look at the diversity of things. I'm not saying living things this time, but let's take a look at the diversity of viruses that you might be familiar with. So the herpes viridae virus. So this one you might know causing cold sores and genital herpes, as well as chickenpox, and for those older individuals can cause shingles. Next, essentially the human papillomavirus. You may have heard of the HPV vaccine. So both males and females can carry HPV, and both should be inoculated so that they have immunity to HPV. HPV is one of those viruses known to cause cancer. Some viruses on the bottom: poliovirus, rubella, West Nile, Ebola, and a variety of influenza, and HIV. The other thing we see in this table is at the first top of the table is the DNA viruses, and the ones at the bottom are RNA. So remember how we said they are one or the other, they're not both. Okay. So for many years in taxonomy, and this isn't just about viruses, essentially things were named based on maybe who first discovered them, or it could be essentially the disease they caused, or some kind of anomaly that they caused. But essentially, even in virology, or we're thinking about the International Committee on the Taxonomy of Viruses, that they're really honing in on a classification scheme that makes sense. Hosts and diseases they cause, okay, that's good. But when you go to other countries, they speak a different language, and they may cause that or call that disease something different. So different, names, common names, like different common names for birds, for example. What does make a lot of sense is naming them based on structure, chemical composition, and their similarities in their genetic makeup. So this really is where taxonomy is going, is stick to that which we can identify and understand that there is some similarities in genetic material. All right, let's take a look at the viral components. So it's super interesting that the viral structure is composed of these regular repeating subunits that give the virus this sort of crystalline appearance. So if you look down below, the one on the left is a naked virus. So you can see all these repeated pieces, if you will, of that virus. On the right-hand side, we see that this particular one is enveloped, so it's not so easy to see all of those. They call it the capsomere, but essentially you don't see that crystalline structure. It's covered by an envelope. So nucleocapsid, two parts. There's a nucleic acid, which is the DNA or the RNA, and then there's essentially the covering, the capsid. The repeating units we'll see later is called capsomere. The envelope then is this lipid, mostly lipid layer that's going to be outside that capsid. So some slightly, some differences there on how they get that. I can come back to that. And then lastly, you can see that both of these viruses can have spikes. Okay, these spikes are going to help the virus essentially attach to or dock to and invade host cells. So you have a nucleocapsid, which is the nucleic acid and the outer crystalline structure. Then we have the envelope for some of those. So they're either naked or enveloped. So some of them have the envelope, and then they all can have spikes. Believe this is a repeat of the previous slide, but I do want to mention a couple of things. The capsid is the most prominent feature of the viruses, and it's going to help us identify or separate them into two groups. So we have our helical types, which are going to look long and narrow like rods, and then we have the icosahedral, and then we're going to have a far different structure. So we can see there's two different shapes of capsid. And the capsomere is that crystalline structure that we refer to when we have a naked virus. pick up